I work on flower development and trying to understand specifically how flowers fabricate features that are really important for plant fitness, whether it's because these features help flower communicate with pollinator or simply deal with environmental stress like lack of water or high level of UV. I've always worked with flower. I started my career looking at genes that control when and where flower form. But I also always like evolution. And really, development and evolution, they go hand in hand. If you want to create something new during evolution, you need to change the developmental process. So this is what we are looking at. And specifically at the moment with my group, we try to understand how plants form patterns on their petals. So I enjoy many aspects of my job. I would say most of them. Still, the most enjoyable aspect is the original thing I found exciting first time I step in a lab, is looking at questions that have been there for a long, long time and figuring out things that nobody had figured out before. When you find a new gene and you understand what it does for the first time, when you see plant doing something you've never seen them do before, this is super exciting. Since becoming a group leader, I must say, go beyond that. I really, really like the interaction with people and the fact that you can help people moving to their next career step. And it's not often you feel you've done this kind of contribution. So contributing to new science is great, but making a real difference in the career progression of somebody is equally great. I think it's favorite anecdote about me doing science simply because it's something I remind myself of all the time, and I found it really useful to lead a group. The first time I stepped in the lab, people gave me a pipette, and I remember thinking, I will have to clean the inside every time. And then 5 minutes later, they bring me the tip, which are the little plastic bits you put at the end and you can throw, so you don't need to clean the inside of a pipette. And I felt really stupid at the time, but now I always remind myself of this because it tells me when I got new students coming, there was a time I didn't know what a pipette was, I didn't know how it worked. And all this knowledge we accumulate, we don't take it for granted. I only have it because people talk to me and explain to me what to do, and now that's my job to do that. General advice for somebody that starts natural science degree: go to as many lectures as you can, attend everything. Don't assume that something may not be interesting. My best lecture were often the one I thought were going to be boring. So you never know. Try everything. And to get into plant science, you will probably have to battle a bit because we get much fewer plant lecture than animal. It doesn't matter. Because actually, plants are really good model system to answer questions that apply across the board of biology. So everything, even if you love plant, everything you learn in animal lecture will be really useful, and you'll be a better rounded scientist because you'll have this duality of plant and animal knowledge. I always like plants, and I think not so much before because we rely on them. I come from a farming family, so I know we rely on plants for our food security, but just because I found them really alien, and I know they are alive like us, but they do life in a very different way. They can poop oxygen and build themselves out of thin air and a bit of sunshine. It looks like they don't move, but actually they move a lot. And I just find their alien experience of life really interesting. So there is lots of exciting new challenge in plant science, especially plant development, which is more my field. One challenge we're trying to tackle is, do we understand enough of how plants use genes to fabricate features that we can now program them? So it's more synthetic biology, generating genetic circuit that you can put in a plant to get it to do whatever you want and to respond to the environment in a smart way. So that's one big challenge. The other big challenge I'm really interested in is we study plant gene and protein for a long time, but now we realize lots of small molecule metabolites are really important for plant development. And we're trying to understand how does that work? What is this link? How do these small molecules are having such a big impact? And it's lots of unknown biology, lots of room for discovery. Metabolites, it covered all sorts of things really. It means that either it can be primary metabolites, so molecules we need like sugar and lipids that are really important for functioning, or what we call secondary metabolites, so more fancy molecules that are dispensable, so they can be molecules that plants use to be colorful like pigment. They could be molecules that can be used as neurotransmitter in animal, but plants make them as well. So it's small signaling molecules that are likely to be perceived by the plant and trigger a cellular response.